Hello everyone, my name is Vineet Chandane and this is a review on the paper Vibrations as a Pitfall for Pyrosequencing. The following paper has been published by Konrad H and their team and is accidental discovery that they made during the study of DNA methylation. They used samples from human blood and human saliva samples, and the following paper focuses on the impact of vibrations on pyrosequencing and the method to curb the same. So before we go into the paper, let's go through some of the terms that they've used in this paper. For example, DNA methylation. DNA methylation refers to the addition of methyl group to the cytosine group of the DNA, which leads to deactivation or turning off of certain genes. This process can be used for certain genetic studies and identifying certain conserved sequences. Methylation-specific PCR is the way to study the phenomenon. And here the DNA is treated using sodium bisulfate to convert the non-methylated cytosine groups into uracil groups, and which during the PCR amplifications are converted into thymine residues. And methylated cytosine remains unaffected. Pyrosequencing is a method of sequencing the DNA using the biomolecule luciferin and the enzyme luciferase, which are obtained by fireflies or certain bioluminescent mushrooms. In this process, the DNA is synthesized and the DNTP attaches to the 3' region of the DNA, and the phosphate group is released in the form of pyrophosphate. The pyrophosphate reacts with the enzyme luciferase and luciferin to give out ATP, which then emits light, which is recorded. After the reaction ends, A-pyrase comes into the picture and destroys any remaining DNTP, concluding the reaction, stopping the emission of light, and then the cycle is repeated. So for the experiment, the scientists used the ProMark Q48 AutoPrep, which is an automatic pyrosequencing machine, and an anti-vibration table. So during the research, the scientists used 3 different setups, one which had only one instrument, in which they ran 130 samples, out of which 42 samples showed a passed result, 59 showed a checked result, that which means that the sample might have failed, and 29 showed a failed result. So to essentially decrease the time taken for the experiment, they added 2 instrumentations side by side, and this led to only 17 out of the 130 results to pass, 16 out of the 130 results to check, and 97 out of the 130 results to fail. The scientists checked what had happened during the experiment to figure out what might cause the problem. After coming to the conclusion that the problem might be due to vibrations, the scientists essentially bought an anti-vibration table and set up the same experiment again using the anti-vibration table and 2 instruments. This led to the finding of the pass samples of 115, check samples 12, and the failed samples were considerably low at 3. This concluded that the vibration can cause hindrance in the result of pyrosequencing and can lead to irreproducible results. Using an anti-vibration table gave them better stability and led to better results. The manufacturer of the instrument should introduce these points in the instruction or create a mechanism that reduces the impact of vibration. And finally, more research is required to better understand this phenomenon. Thank you.